You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health Podcast. I have Dr. Peter Christey. He's the founder and CEO of GALT, General Automation Lab Technologies. And they isolate, cultivate, and screen the living microbiome, thousands of microbes at a time, which is really, really cool. It's a high throughput way to look at microbes, it looks like. So, Peter, thank you for coming.
Well, thank you. Thank you for having me.
Yeah, if you would, tell me, how did this idea and methodology and all that come about? How did the whole company come into action?
Well, that's a really interesting question. So about 4 or 5 years ago, a few of us got together to think about this problem of microbial cultivation. There was a number out in the literature that less than 1% of the microbial species that currently exist on the planet have ever been cultivated in the laboratory, and that was proving to be a real barrier to development of microbial-based products. So at that time, there was a lot of interest, particularly in the ag tech arena, around developing microbes for use to activity of crops and usage. Mm-hmm. But one of the barriers was that when potential microbes that they wanted to develop into products, the whole cultivation process was incredibly slow and cumbersome and a real barrier to progress in this field. So we got together and said, hey, what can we do to solve this problem? Because if we could solve it, there would be a massive amount of benefit from that. And so we sat down and worked out a way to improve cultivation rates and developed the platform that we have in the company now.
When you say cultivation, do you literally mean cultivating a given organism and getting enough of it to study, or do you mean sequencing?
So our primary goal is cultivation and getting a pure isolate into culture in the lab so that you can then evaluate it for a product candidate or else study its biochemistry and physiology in a lot more detail. Some of that downstream study may involve sequencing, but our prime company is to deliver pure living isolates of interest to the researcher or the product developer.
And forgive me, an isolate is the whole organism, or is the isolate a particular metabolite of an organism?
So an isolate is a pure or a living organism. So, for example, if I have a strep infection in your throat, and I take a swab and I want to understand more about your particular strep infection, I want to get pure living isolates of that infection growing on a petri dish or in a 96-well plate in the lab so I can study it. So the goal there is to get a living colony or a living culture that's pure of the organism that we're interested in, and whether that's a strep strain, whether it's a nitrogen-fixing bacteria isolated from a sample, or whether it's a butyrate producing bacteria in the human gut. The goal here is to get a living, pure colony or population of organisms of interest so that you can study in the laboratory.
So is it harder to cultivate certain strains at all, or is it harder to isolate one particular strain and not have contamination of other strains?
Yeah, I mean, this is a key question. Like, there are microbial, like if we take a soil sample, for example, there are certain microbes in soil that are very, very easy to grow. They tend to be the fast-growing organisms. And organisms that can live in many, many different conditions and environments. And those types of organisms are very easy to grow, and they've been growing regularly, in labs over many, many years. But often the most interesting organisms are the ones that may be less abundant in a soil sample, or they may be slightly slower growing in that sample or in your laboratory system. And so those ones, the ones that are often more interest, are sometimes a lot more difficult to grow. And those are the ones that our system is designed to help researchers get more access to so that they can grow them easily in their laboratories and then study them.
Yeah, it's funny. I just, I don't know. I'm not in the industry, but I just assumed it's easy to cultivate bacteria and you don't even have to do anything. But the sequencing is hard. I didn't realize it was a challenge to do it.
No, it's interesting is that if you, obviously, people who are listening to the podcast may be very aware of the importance of the microbiome, particularly in human health. And how it's been connected to many, many different disease states like diabetes and metabolic disease and autoimmune disease, allergies, Parkinson's disease, the list goes on and on. And as a result of that, people are very interested in studying the microbiome. Human gut microbiome and the human skin microbiome are areas of a lot of interest. And the primary method that people are used to date to study those microbiomes is actually DNA sequencing, because it's relatively straightforward now to take a human gut sample or a human skin sample and the mixture of microbes in that sample and then sequence it. And that sequencing will give you some information about what microbes are there at that particular point in time. So that's actually what's driven a lot of the scientific insight that has developed in the world of the microbiome over the last 5 to 10 years has actually been sequencing based. The challenge has been and still is that if those sequencing studies identify a microbe of interest that you may think may be involved in a particular disease mechanism, for example, the difficulty is how do I get a living culture of that microbe in a pure state and into the laboratory so I can study it in more detail and really validate that that microbe is involved in the disease state, and if so, how is it involved in the disease state? So it is going from this concept of, okay, sequencing tells me there's a correlation between this microbe and the disease. How do I go to causation where I show that that microbe is actually involved in that disease state? And that's where you can start, once you've validated that and understand the mechanism, you can start to think about interventions that may be based on those insights.
So what are some of the methods to culture bacteria that you guys are using, without giving away proprietary stuff, but what are some things that you found are necessary to culture bacteria in the right way, keep them alive?
Well, and I think it's useful to provide a little bit of context about what's been going on in the microbiology lab in the last 50 to 100 years. Is microbiology is still using tools that were developed in the 1800s. For example, the most common device used for cultivation in a microbiology lab is the Petri dish, and I think most people will be familiar with that. That was invented in 1887, and another common lab tool is the Bunsen burner that you see in just about every microbiology lab today. That was invented in 1855. So in microbiology, the available tool set to cultivate and study complex microbial populations is over 100 years old. And this contrasts very markedly with other fields of biology like genomics. If you go into a genomics lab, you'll see a lot of very, very sophisticated analytical instrumentation that's been developed to help study the genome. And in cell biology, you see a lot of very sophisticated instrumentation such as flow cytometry, high content screening, and confocal microscopy that's used to study cells. But for some reason, the microbiology field has not benefited from the development of these type of tools. So what we're doing here at GALT is developing a instrumentation-based tool set to greatly facilitate the whole cultivation process in microbiology labs and greatly increase the productivity of that process. So the core technology of our platform is a microarray that has fabricated into the surface of it many thousands and thousands of tiny little growth chambers. So this allows us in a very small device to cultivate in parallel an incredibly high number of isolates of pure bacteria on the device. And then the instrument that we developed transfers interesting targets that grow on that device into a 96-well plate, which is the standard format in the lab. So the benefits of that from a efficiency and a productivity point of view are such that it opens up whole new areas of research strategy that weren't really accessible before because they were too cumbersome and too slow.
Why is it better to culture a bacteria in a bunch of wells instead of one big dish? Is it because each has its own food source that's not competed for by other bacteria?
Well, I think that there's three major elements that we were looking for when we designed this. So one is throughput. Typically a petri dish, you can harvest between maybe 25, 50, maybe 100 colonies off that petri dish. That's okay if you're studying a single pathogen, back to the strep infection example that I used earlier on. If you just want to grow one single species, that's fine. But when you are studying a complex ecosystem, which is the microbiome, which may contain many thousands of different species in them, you really need a tool or a device that has a far higher sampling rate. So the first key aspect of the system that we've developed is a very high sampling rate. The second key aspect is that our system prevents the device being overrun by fast-growing bacteria. So on a petri dish, you'll often get overgrowth within 24 to 48 hours of fast-growing bacteria that essentially obliterates slower growing and potentially more interesting bacteria. So that is a facet of our system that doesn't exist. And then thirdly, we use a liquid culture format for our system that preferentially grows bacteria that prefer liquid culture rather than a solid agar surface. So these are three technical aspects of the system that make it to grow more difficult to grow.
Are there are there certain strains that you've been able to cultivate that no one else has been able to because of your conditions?
We have over time cultivated a lot of bacteria on our systems, and some of them have not been identified before. I don't have any numbers around that, but definitely our system has identified and cultivated things that don't show up in the standard databases.
That's pretty cool. And when you have optimal conditions,
that is cool.
But when you have optimal conditions for the bacteria, how much faster and more robust have the colonies been that you've created?
The robustness of the colonies is such that I think once you get a you've cultivated a bacteria in a 96-well plate. I mean, if you have a species, you have that species, and it's not more robust or less robust on our system. The key thing is that if you want to study that bacteria, you have access to it a lot more quickly. And I think, one example of a clicking with is someone who's studying a very slow-growing bacteria called actinobacteria. And to support their research, they wanted to develop a strain collection of many, many different species of actinobacteria so that they could studies and test them in their research. So before we arrived in their lab, they'd been working for about 2 years to develop a strain collection of 400 different species, and they'd been using petri dishes, which was a very slow and arduous process. Once they started using the Prospector system, they were able to develop a second strain collection of 600 within 3 months. So that provided for them a tool set, which is the strain collection, to support their research and they were able to develop that tool set far faster and far easier than what they could do with current technologies. So this is the difference between taking 2 years to get to where you want to go to taking 3 months, which is a major advance if you're studying a microbiome-based disease, or if you're an agricultural biotech researcher looking for microbes that can enhance crop productivity, for example.
I would think you'd have to see different bacterial behavior if it's cultured in wells versus just a dish. I mean, maybe it prevents the formation of biofilms, encourages them, or you have like a certain amount that can fit in a well, I guess, maximally. Maybe bacteria are like fish and they grow to the size of the well, or they proliferate to the size of the well. I mean, what interesting things have you seen that are different with your method?
Well, I think, that's a good point. So the end result, as I was saying, is, you either get an isolate or you don't. And the end result is kind of the same, whether we use a petri dish or an array-based approach. But you're absolutely correct that on the array-based approach, things grow differently. So, one example of that is that we find that we're able to detect growth on our array far faster than what you can on a petri dish. And the reason is that the microwells that we're growing these microbes in are about 3 nanoliters in volume. So bacteria growing in that microwell, you can detect them quite early in the growth phase. And whereas on a petri dish, you have to wait perhaps 3 days because the bacteria have to grow into a visible colony on the surface of the petri dish before you can see them. So we're actually exploring this to understand in what situations would it be a benefit to be able to identify growth of bacteria time or a third of the time versus a petri dish, and where would that be beneficial? So that there's certain applications and potentially in the medical arena and diagnostic arena, we're getting faster results may be useful, and we're looking at those to try and understand that. In terms of why another way that they grow differently on the arrays is that, and I referred to this before, is that because we seal the growth chambers with a membrane, bacteria can't outgrow their growth chambers and take over the entire array, so that fast growers are trapped in their tiny little growth chambers, and this allows time for slower growing or more fastidious bacteria to grow up in their own microwells and then be seen on the array. So this is why our collaborator that I referred to before was able to detect and isolate a big strain collection of slow-growing bacteria relatively quickly, because on our array format, his slow-growing bacteria weren't being rapidly overgrown by fast growers, which is what happens on a petri dish.
Well, within a given strain, though, is there literally individual behavior of different bacteria all in the same strain, or is it just across strains?
So could you just clarify that question a little bit, Richard?
Yeah, sure. Right. So if I have one particular strain of bacteria, do all the bacteria grow at the same rate, or are there fast ones and slow ones? When you were just talking about fast and slow, did you mean within the same strain, or comparing across two different types of strains?
No, these are typically different species or different strains, depending on what.
Okay.
So, for example, in soil, so a typical soil sample may have 1,000 different species in it. Some of those species are very fast-growing species. They may be motile, they may swim around, and they may be highly abundant. And one typical example is Pseudomonas, is a very common soil bacterium that's typically anywhere between 10, maybe 20% of the microbes in the sample could be Pseudomonas. They're very fast growing, they replicate quickly, and they swim around. So you put these onto a petri dish, and within probably 24 hours or 48 hours, your entire petri dish is just completely overcome with Pseudomonas. And then there may be other bacteria in the soil, kind of species number 732 out of the 1,000 that may only be present at 0.01% of the total bacteria in a sample. They may be slower growing. They may be non-motile. And if you plate those onto a petri dish, you're never going to see them. Because they just have very, very different dynamics from, say, a Pseudomonas, and they're not going to be cultivatable because they'll just simply be overrun. So on the array-based format, it's a lot easier to grow and isolate those slower growing components of a complex microbiome because of the way the chip is designed.
Yeah, see, again, I just assumed you were somehow able to isolate one particular strain of bacteria, put it in the microwave and grow it. But what's the reality of it? Can you not isolate first and then grow? Or is there, for some reason, you have to depend upon the geometry and the special elements of the microarray in order to separate out growth?
So what we do is, the way the array works is we take a sample such as soil, and then we isolate from that a suspension of microbes from the soil sample. And then we dilute that into a medium that we want those microbes to grow in. Then we load that suspension of microbes onto the array such that individual microbes partition into growth chain. And then we seal it, and then the individual microbes grow up. So what we have on the array is hundreds and hundreds of microisolates in their growth chambers that are all derived from the original microbiome sample, and they're different because they're all derived from a separate microbe in that sample. So the array itself will have a large number of isolates that represent a multitude of different species from the microbiome sample that you first started with. And then, so we've essentially digitized that microbiome sample onto the array. And then we're able to go in and select microwells that contain isolates, and then take them into a 96-well plate for downstream testing. So what drives what microbes grow in the array? Typically, it's what is the original microbiome sample and what bacteria were present in that. And then the second major element is the medium, the growth medium that you choose to load onto the array with the bacteria. So with different growth media, you'll get different cuts of the microbiome and different subpopulations of that growing on the array.
What if you only want one particular strain of something you separated out before or after it?
So if you're looking for a specific target, there's a couple of ways of doing it. So one is to take isolates from the microarray and put them into 96-well plates, and then do a downstream screen So, for example, if you have a lot of people do DNA sequencing. So if you have a 96-well plate of isolates, you can do Sanger sequencing on those isolates to identify what they are and then select the one that you're interested in. Another way of selecting for what you're looking for is to do on-array screening. So if you have a fluorescent reporter system, for example, for a particular enzyme that you may be interested in finding an isolate that secretes an enzyme, you can put that reporter system on the array and then use the optics in our instrument to read the array and identify microwells that contain what you're looking for. A third approach is to use selective media on the array so that when you take your complex microbiome sample and load it onto the array, you load it with media that only support the growth of your target bacteria, and then the colonies that grow up on the array would be highly enriched in the targets that you're looking for. So depending on the system you're working with and the kind of scientific questions you're trying to answer, you can use this system in a variety of different ways to get to where you want to go.
Have people, or has anyone profiled specific bacteria, what they love to eat, what they don't like to eat, how fast they grow, what metabolites they produce, what phages attack them, et cetera? Does anyone have a workup on a given bacteria so we know as much as possible about it?
Using that's a potential application of our system in the future. We're not focused on that. We're focused on delivering living isolates. But in terms of, separate from our system, obviously in the world of microbiology, a lot of microbes have been characterized once they get into the lab environment and once they've been isolated as a pure colony. You can take them downstream into the lab and do those analyses. But again, and that's been done a lot with bacteria that are easy to grow using petri dishes. So what we enable is, the ability to get more challenging bacteria to cultivate and to be able to go downstream and do those same kind of characterizations.
And when something's an isolate, what percentage purity is it, and what can be mixed in with it?
We typically get very pure isolates coming off the system. I mean, because of the way we load the chips, a small proportion are actually mixed colonies, and that's just a natural outcome of the mathematics of the way we load these chips. But we get typically very pure colonies coming out, and we demonstrate that by doing DNA sequencing on the isolates. And so they're able to take those downstream and do their studies on those pure isolates.
The reason I ask is in some worlds, 99.9999% is, achievable and that's great. But I just wonder in the isolate world, what's a high purity level versus a low one?
So we use Sanger sequencing to assess purity. So, I could go into the mathematics of that. In terms of, that's a good question. I'd have to think about that a little bit more. What an absolutely pure isolate, it starts to become a definitional thing at that point, because as colonies grow and develop over time, there will be some natural variation that comes up in a culture of bacteria. When you bring a culture up, not everything is going to be exactly the same, because you get genetic drift as the culture grows and develops, and as you get billions of microorganisms evolving as they do over time. So then it becomes a definition of what do you mean by purity. But typically, when you're working with systems that are above 99% purity, for what you're looking for, that's very adequate for most of the experimental work that's been done.
Okay, that's what I was wondering. I just didn't know how pure pure was. So, have you seen any different behaviors of any of the bacteria once they are purified to such a high degree? Do they act differently versus when they're around all kinds of neighbors?
We haven't observed that primarily because we're focused on developing our system and cultivation, but it's well known in the field of microbiology that microbes often act within the context of the community and the environment that they live in. So bacteria sometimes can do things with other bacteria that they can't do purely by themselves. And some bacteria, it's known that they actually require the presence of other bacteria in order for them to be able to grow. So, for example, they may require some nutrient, or they may require some growth factor, or some other feature of the presence of another bacteria in order for them to be able to grow. And so these are called syntrophic or symbiotic relationships. So that's a feature of these complex microbiomes. And this goes back to the point why it's so important to get living isolates to study in a laboratory, because you can start to dissect these relationships and the functionality of microbes a lot more specifically when you have pure isolates in the laboratory, and you can mix groups of bacteria together and study how they function as part of a larger community, and understand, in particular disease states, what combinations of bacteria may be important versus single bacteria. I mean, this is a really complex world that we live in, and the benefits of understanding all this are going to be so huge that having the tools to support these kind of experiments is absolutely critical.
Have you had any bacteria battles where you culture two isolates and then put them in the same dish and have them battle it out and see what happens?
No. That's a really interesting concept, and I'm sure we'll get to it one day. But we're absolutely focused on getting our system, developed and out the door, and starting to get to the point where we can ask these type of questions and say, hey, we've got this whole new platform for doing microbiology. What are the cool things that we can do with it? What are the things that we can do with this system that we couldn't do before because the system never existed? And some of the things that you're suggesting sound like great ideas.
Oh, good. Are there any dream bacteria that you'd like to culture but are so far impossible for some reason?
We haven't identified the dream bacteria, but there's kind of obviously areas of interest in different fields that we work with. And one example is in ag bio. There's a lot of interest on identifying bacteria that can fix nitrogen. Nitrogen fertilizer usage in agriculture is being hugely beneficial in terms of helping feed the planet and raise the productivity of the way we grow crop plants. But it has its downsides, and the downsides are that nitrogen fertilizer can have negative effects on the whole ecosystem by kind of causing, algal blooms and a whole bunch of other stuff that's not great. And also the production of nitrogen fertilizer is incredibly energy intensive. I mean, there's a good proportion of the energy usage today across the planet is used to generate nitrogen fertilizer for agriculture. So there's a lot of people really interested in finding microbes that can fix nitrogen from the air, and so potentially allow us to reduce the usage of nitrogen fertilizer globally. So, if our system could help find that magic nitrogen fixing microbe that could, really reduce significantly the amount of nitrogen fertilizer that's used, that would be amazing, and it would it would be fantastic.
Yeah, perhaps your technology could be used to bacteria that are in very, very low amounts in a given system that are usually invisible because of the other bacteria that predominate them.
Mm-hmm. Absolutely. And I think that there's other disease states, I've talked about agriculture, but if you go into the human gut, and there's kind of connections being drawn between major diseases like diabetes and the microbiome, metabolic disease in the microbiome. There's been some recent papers come out that's shown or indicated that the outlook for patients that have pancreatic cancer may be driven by the microbiome that exists within their tumor, for example. So in any one of those disease states, as we delve into it deeper and understand the mechanisms by which the microbiome has these effects, being able to identify and cultivate the specific 1, 2, or 3 bacteria that are critical in those diseases, and then be able to use those bacteria as therapeutics or as treatments for these patients would be an incredible breakthrough as well. So using our system to find and isolate those bacteria would be an incredible achievement for us.
So I guess in our guts we have what, like how many different strains are thought? 1,000, tens of thousands?
Again, it becomes a definitional thing, but I think in a typical human gut sample, you'll have anything from say 300 to 600 different species. And then within that, there may be different strains and subtypes and such like, and that those species will vary from one individual to another. So you have to look, from a population perspective versus an individual perspective. But yes, these are the complex ecosystems that do a lot in our gut that I think we've only realized how important that is in the last 5 to 10 years.
So what's your goal from here forward for the next few years? Do you want to just get faster at cultivating? Do you want to also cultivate uncultivatable previously strains? I mean, what would be the big wins for you guys over the next few years?
Well, the big wins, I mean, there's 2 levels of looking at this. The big win is, from a technical perspective, is cultivating faster and cultivating, previously uncultivated ones and cultivating bacteria that, make a real difference from a health and human wellness perspective or from an agricultural perspective. But, the real goal of the company is to provide the tools to the microbiome research community that will accelerate this field by 1 to 2 decades. I mean, I've seen enough evidence in the literature and some amazing studies that have been done out there now in a variety of different disease states that really point to the fact that the microbiome is a really critical area of science that we need to understand in a lot of detail. And when we understand it, there will be a huge amount of benefits to humankind from that understanding. And that understanding will be applied in human health, wellness, agriculture, and a large number of different arenas. So that us company, if we can help the researchers make their insights faster, by providing tools that really solve problems for them. That's the goal of the company. And the current platform that we have now is a huge step forward in that direction. And currently we're focused on cultivation, but there's a lot of other different applications we can layer on that in the future. But I'm really excited about, potentially developing other technologies that solve different problems in microbiome research, or bringing in technologies from other areas of life sciences and applying them in the microbiome research arena, all with the goal of really accelerating this enormously important field of science. I mean, I think that for the last few years, the microbiome research arena has been thought of as a separate area of biology. But now we realize it's so connected to everything we are as humans and what we do that it's not a separate area of science. It's really an integral aspect of understanding human biology. So as we go forward, anyone who wants to understand human biology or study an aspect of human biology is going to have to take account of the microbiome as they go through their study protocols.
So do you find it hard to stay on target and just be a microbe farmer and not want to delve into all these other things?
It's obviously a temptation. There's so much, so many directions we could go, and you're staying on target. It's always intriguing, all the places we could go, but we're very focused. And I think some of the questions you asked, earlier on in the discussion about have we done this, obviously we've thought about many of those things, but currently we're just focused on getting our product developed, getting it out the door, and starting to have an impact so that we can build from that.
Well, one last thing I thought of, if you don't mind. I imagine someone taking, I don't know if this is possible, but taking one of your 96 cell microarrays and, putting 4 different nutrients or chemicals or whatever on each of the arrays to create 4 different environments, and then taking it to, like, let's say a mine where, an effluent is coming out, and sampling the effluent that's coming out. And I could probably quickly see amongst the 96 wells, if I had 4 different environments created in the wells, what bacteria would tend to grow there. Maybe that would help me assess what's going on in a place like that faster. So maybe your technology could be used out of a lab like that.
Absolutely. I mean, those are the kind of applications that we look forward to developing. And just to level set here is the typical technology used in a lab. A petri dish is like 3 inches across, and then a 96 well plate has 96 different wells on it. Our array that we use is 1 inch by 3 inches, and it actually has 6,000 microwells on it. So when I was talking before about having a fundamental increase in the sampling rate and the throughput on our platform, that's the kind of order of magnitude differences that we're implementing here. So, there's a potential to have, not just 96 different things on the array, but, thousands. And that opens up a whole new world in terms of what you can do with the system.
Yeah, that's great. Well, excellent. Well, thanks for taking the time. What's the best way for folks to get in contact or to ask questions?
Feel free to email me. So my email address is Peter, PETER, at GALT-INC.com.
Very good. Well, Peter, thanks for coming on the podcast. I really appreciate it.
Super. Thanks, Richard. Thanks for the opportunity.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.